Hi students, welcome back to our long lecture series on hemoglobin. This is the final video lecture where we will be summing up, summarizing and completing the entire competency on hemoglobin. So, these are the two competencies. Okay, we have discussed in detail about competency 5.2. So let us recap. What did we discuss? The competency says describe and discuss functions of proteins and structure function relationship in relevant areas, right? Hemoglobin, they give a specific example and selected hemoglobinopathies. Okay. They also, NMC, National Medical Commission, NMC 6.12 also says describe the major types of hemoglobin and its derivatives found in the body and their physiological and pathological relevance. Okay, so we already completed this in great detail, right? And we also dealt with this where we have discussed embryonic hemoglobin, we have discussed fetal hemoglobin, adult hemoglobin, all right, and various types of adult hemoglobin, HbA1, HbA2. We also discussed HbA1c, right? That is a modified type of adult hemoglobin that depends on your blood glucose concentration. Also, we discussed abnormal hemoglobin variants like carboxyhemoglobin and methemoglobin, right? So we will directly continue with our abnormal hemoglobin variants. So these are the few abnormal hemoglobin variants that we discussed, right? I also told you most important among them will be three: this carboxyhemoglobin, methemoglobin and sulfhemoglobin. Alright, we already discussed carboxyhemoglobin and methemoglobin in much detail in last video lecture, and today we will continue with sulfhemoglobin. Okay, so what is sulfhemoglobin? As the name suggests, it's simply nothing but sulfur. Sulfur gets linked to hemoglobin anyhow. Alright, so how sulfur can get linked? Okay, so what is the source of sulfur? For sulfur to get linked, we need sulfur. Sulfur अगर जुड़ रहा है hemoglobin के साथ तो sulfur चाहिए. So we can get, the body can be exposed to sulfur in many cases. Most common are ingestion of sulfur containing drugs. There are many medicines or tablets or whatever you want to say, drugs that have got sulfur compound in them, right? Of course, you will be learning their names when you go to second year or pharmacology classes. But for now, just know sulfur containing drug is one such event or condition that may lead to formation of sulfhemoglobin excess ingestion, right? And also, in case of chronic constipation, what happens in chronic constipation? This we know, right? Every one of us is passing flatus knowingly, unknowingly, willingly, unwillingly, right? And most often that is odorless to a great extent. But if someone is having constipation, बहुत दिन से नहीं गया है तो बाकी लोगों को घर से बाहर निकलना पड़ता है ना? Because the bacteria in the intestine acts on unexcreted food material, or that is material present in large intestine, and they produce H2S. Right? This H2S, if it is produced in excess, is also another source of sulfur that may lead to formation of sulfhemoglobin, right? Now, sulfhemoglobin is a greenish compound, okay, where very important, sulfur is attached covalently to porphyrin ring, not iron. This is a very important MCQ. All the other variants, most commonly, they target the iron, right? However, in spite of the fact that iron is free, sulfhemoglobin cannot bind with oxygen because it changes the structure. It leads to a steric hindrance, right? Anyway, your important point to note is, in case of sulfhemoglobin, the sulfur is covalently bound to porphyrin ring. It is not bound to iron, right? However, the problem here is, since it is covalently bound, okay, this is irreversible. Once a sulfhemoglobin is formed, it cannot be reversed by increasing oxygen concentration, right? We saw in case of carboxyhemoglobin, giving excess oxygen reverses the carbon monoxide poisoning. In case of methemoglobinemia, giving reducing agent like ascorbic acid, methylene blue, all those actually can convert back the methemoglobin to hemoglobin. So these are all reversible. but formation of sulfhemoglobin is not reversible. Okay, so once it is formed, the hemoglobin is rendered non-functional. So here are a few drugs. So if you are actually not a first year student and you are watching this video anyway, or you are preparing for NEET PG or MCQ, you should know a few common drugs that contain sulfur containing drugs, okay, that actually produces sulfhemoglobin if they are taken in a toxic amount. So number one is dapsone. First year students, again the names will appear Hebrew, okay, because you don't know drugs right now. So you should know this is the leprosy containing drug, okay, Microbacterium lepri, okay, hota na code. So dapsone, this leprosy treating agent, phenacetin acetylenolide, sulfonamide. So basically just ratta marlo or just know these names if MCQ comes. To be honest, there are multiple hundreds of sulfur containing drugs, but there are 4 to 5 drugs whose names you must know, because MCQ question will come as all except, okay, all of these drugs can produce sulfhemoglobin except. Then you need to select the medicine. For example, paracetamol is given as an option, that doesn't contain sulfur, okay, and that will not produce sulfhemoglobin. So basically you should know few drugs being a first year student. Paracetamol is one such example, alright. Paracetamol, atenolol is a drug that we give for lowering blood pressure, okay. So few basic drugs you should know. Metformin is one such drug that we prescribe for lowering blood glucose, okay. L-troxin is one such drug that we use to treat hypothyroidism. Anyway, I will give you, when the class resumes, believe me, I will give you some names of drugs that will be very handy for you to prescribe. Just remind me when the class resumes, okay. Anyway, so this was all about abnormal hemoglobin variants, okay. I intended to complete sulfhemoglobin in last lecture, but it would have been stretched the already long lecture anyway. So sulfhemoglobin is over. So if it comes as a viva, fine. I don't think it will come as a short note, but the few things are important for MCQ, that is iron is not bound, porphyrin ring is bound to the sulfur, and it is irreversible, okay. So now myoglobin, alright. So hemoglobin is over. Now we move on to another hemoprotein, that is myoglobin, okay. So again we will take up myoglobin just like we took hemoglobin, but in much less detail, okay. We spent 6 to 7 lectures on hemoglobin. We will spend hardly 20 minutes maximum on myoglobin. Anyway, so myoglobin is a hemoprotein of muscle, okay. Mainly it is found in red muscle fiber, okay, red meat that we call. So however, primarily the main area or the main muscle group where myoglobin is in excess is cardiac muscle. Of course, it is also present in skeletal muscle, but the amount of myoglobin present in cardiac muscle is much higher, okay. You will understand why very soon. Mainly because myoglobin ensures that the tissues, the muscle always gets oxygen, okay. And what muscle in our body needs absolute surety that you should get oxygen? That is the heart, definitely, because other skeletal muscles have got a room for resting. Even when you are sleeping, when you are just sitting, I am sitting, my biceps are generally not working that much hard, okay, and my lower limb muscles are idle only, but my heart is beating. Your heart is also beating, hai na? Dil dhadakne do. So heart muscle always needs oxygen. So in case of hypoxia, mild hypoxia, our muscles can get away with it by lactic acid, etc., but heart muscles cannot, because it is always functioning at the top level. So ischemia, infarction, all those things occur. So to prevent that, nature has given a basic protection to them. Even if the oxygen saturation is very low, still the heart muscle can get oxygen via myoglobin. So looking at the structure, myoglobin is a monomeric unit, means it has got only one subunit, the globin chain, right. In case of hemoglobin, there were 4, right. So it has got one globin chain and one heme moiety. Moiety means residue or single structure. So this is how myoglobin looks. Definitely it is composed of alpha helix, and you see basically it's a single subunit of looks like a single subunit of hemoglobin only. All right. In case of hemoglobin, there are 4 such. In case of myoglobin, it's only 1, right? So here are a few. This slide is about numbers. You don't need to remember much, but 2 things. Number 1, 17,000 Dalton or 17 kd is the molecular weight of myoglobin, and this is important. It is rich in alpha helix. I already told you hemoglobin and myoglobin are 2 such globular proteins. Okay, it's a globular molecule. Just a minute. It's a globular molecule with alpha helix as a secondary structure. What are these? X-axis, Y-axis and Z-axis. I already told you during hemoglobin discussion, right? Anyway, so spheroid globular molecule 17 kd and mainly alpha helix in secondary structure, right? However, this is important, 153 amino acid residues. This is for MCQ. Again, it is not possible to remember. You need to revise it the day before exam. So, number of residues present in hemoglobin, all the alpha and beta variants of hemoglobin, as well as the myoglobin. These are very irritating MCQs that are often asked, and if you know, you can mark. If you don't know, you can guess. Right? So, being a single polypeptide chain, what is important is hemoglobin was binding with 4 oxygen, right? So, one oxyhemoglobin molecule had 4 oxygen. However, one hemoglobin, since it has got one heme, okay, it is binding with 1 iron and 1 oxygen molecule, right? So, the ferrous iron, mind it, in case of myoglobin also, it is in ferrous form. Of course, it can be converted to ferric form. We will be discussing it very soon. And same in case of myoglobin also, oxygen binds as the 6th ligand to the iron. Okay? Iron is bound to 4 pyrrole ring. Okay, the basic cytochrome structure is the same. Don't confuse the cytochrome structure with globins. Okay, globins are proteins and iron is bound to the 4 porphyrin ring. On one side it will be bound to, in case of hemoglobin it was bound to histidine, and on the other side, on the 6th zone, 6th bond was binding to oxygen. Same occurs in case of myoglobin, but histidine is different. Okay, in the 5th position you don't need to know. Basically what you need to know, this, if you are given a difference between hemoglobin and myoglobin, oxygen binds as the 6th ligand to iron, this point is same in case of hemoglobin and myoglobin. So, examiner, since there are a lot of differences, examiner may try to trick you or fool you by saying, okay, can you tell me what is the similarity between hemoglobin and myoglobin? Of course, there are multiple similarities. Same iron containing hemoprotein, globular protein, secondary structure, alpha helix, both bind with oxygen, and oxygen binds at the as the 6th ligand. Okay, so these are the similarity. Let me repeat so that you can note down. Both are hemoprotein, both are globular protein, both contain iron and globin, both are alpha helix in secondary structure, and both carry oxygen. Okay, and oxygen binds to the 6th position. Okay, if you missed something, you can just rewatch the recording, which I will send you sooner. Anyway, so myoglobin, again we are looking at the differences now, has got a very low, very low P50 value with 2 to 3 torr per millimeter of mercury. We are talking drastically low value. We saw that in case of adult hemoglobin, it was 27. We told, okay, fetal hemoglobin has got a very high affinity. So, how much high affinity was there? The P50 was 22, right? But you can understand, you can visualize how much more affinity myoglobin has. It means even if there is 2 to 3 torr of oxygen, I mean the partial pressure is, even if the partial pressure is so low, still half of myoglobin will be saturated. Alright, so it is extremely low. Therefore, okay, the oxygen dissociation curve is a hyperbola. You need to understand why it is hyperbola. See, over here, how even if adult hemoglobin and fetal hemoglobin, they have got high affinity, the affinity actually falls off when this is HbF, for example, because HbF, the curve shifts to the left, and this is HbA1, right? So in case of HbA1, below 27, at 27 it is, suppose this one, okay, at 27 this is half saturated, right? And below 27 it falls off. In case of fetal hemoglobin, it is half saturated at 22. Below 22 it will fall off. However, we see in case of 2 to 3 torr, when the partial pressure is very low, both adult hemoglobin and fetal hemoglobin will give out oxygen. When the curve is here, basically oxygen affinity is 0. It means oxygen is being dissociated. सारा दे दिया ऑक्सीजन, अगर एडल्ट हीमोग्लोबिन एंड फीटल हीमोग्लोबिन सोच रहे हैं तो. But still, even in that partial pressure, myoglobin is almost more than half saturated. Okay, so if you see, this will be around 2 to 3 torr, and at 2 to 3 torr, hemoglobin, adult hemoglobin, fetal hemoglobin has got almost 0 affinity. It will never bind to oxygen. However, myoglobin still grabs the oxygen, right? So you can ask, sir, then myoglobin is very good in grabbing the oxygen, it will never let the oxygen go. Yes, that is what myoglobin does. It actually grabs hold of all the oxygen and it never lets oxygen go till oxygen saturation falls as low as almost 0. So since it is 2 to 3, when oxygen saturation, I mean the patient is completely anoxic, I mean it is gone, still the heart will continue to beat because of this reserved oxygen myoglobin. Okay, so it's a protection of nature. So in comparison to hemoglobin A and fetal hemoglobin, myoglobin has high affinity for oxygen. I could have added very high affinity for oxygen. Okay, so and that is reversely related to P50 value. So HbF and myoglobin has got low P50 values as compared to P50 value of HbA1. Okay, when you compare to HbA1, both HbF and myoglobin has got low P50 value. However, the P50 value of myoglobin is lower than that of fetal hemoglobin. Okay, so what happens with low P50? More oxygen binds at low PO2. So even when the partial pressure of oxygen is very low, still more oxygen can bind to myoglobin, right? So what are the functions of myoglobin? What does myoglobin do? Myoglobin is found in cytosol of skeletal and heart muscle mainly. This is the main area. In skeletal muscle it is also found, but not as much as compared to heart muscle. So myoglobin facilitates rapidly respiring muscle tissues. So muscle tissues that are working very hard, for example heart, okay, what they are doing? Respiring means electron transport chain, cellular respiration, production of ATP. ATP is needed for muscle contraction. You already read the stretch reflex, how it works, troponin T, troponin, tropomyosin, actin, myosin, whatever. I am not going into the details. So we need oxygen, right? So the rate of oxygen diffusion is same for all the tissues, okay, because blood is flowing at a same rate, right? So it will provide heart via the coronary artery, it will provide muscle via the brachial artery. I am giving an example for biceps. So the rate of diffusion of oxygen from the blood to the tissues is same for all muscles, okay. However, if a muscle is working very hard, okay, that much amount of oxygen will still be insufficient. For example, I am giving you food to eat, okay, I am giving you fixed food, 10 biscuits per day, right, to everybody, to you, me, him, her, everyone. But when someone is working very hard, working out very hard, okay, gym, that will be insufficient. However, the rule is my supply of food is same for everyone. So he needs something so that can catch hold of the food and supply during reserve. Similarly, this, because of low solubility, myoglobin increases the solubility of oxygen. How it increases the solubility of oxygen? Basically, it forms MbO2, or it combines with oxygen and it remains in the cytosol. So instead of oxygen being dissolved, it now combines with myoglobin and it enters the cytosol, remains in the cytosol anyway. So myoglobin facilitates oxygen diffusion. Oxygen is getting bound very rapidly. So instead of going very slow, it is absolutely quickly trapped by the myoglobin. So myoglobin increases the solubility of oxygen. It does not mean that oxygen is getting actually dissolved in water or blood. Okay, basically it forms MbO2. With respect to the oxygen, basically it is going inside the solution. Okay, so it increases the solubility of oxygen and it facilitates oxygen diffusion. So this again I told you is a storing hemoprotein. It acts as a reservoir of oxygen. Whenever needed, myoglobin will supply. This is the purpose of reservoir. We never use forget water. Okay, water का tank में तो reservoir में रहता है, वहाँ से use करते हैं. But generally the thing is reserve force is always in case of emergency. ठीक है? Now you see myoglobin does not allow oxygen to remain in free state. What is free state? That is oxygen in form of O2 molecule in aqueous medium, right? It is there in muscle में रहता है, very low amount, it is less soluble, but still it remains free. In muscle what happens? Oxygen diffused in muscle cells is used up in oxidative phosphorylation. Basically it produces ATP. So in muscles, oxygen has got one fate. It will be used up for ATP synthesis via oxidative phosphorylation. Same in case of all muscles. Okay, so naturally not 100% cannot be utilized. Okay, these are all reversible reactions. So some part will remain free. So in skeletal muscle, that remains as free oxygen that gets transported in the blood, or it binds to hemoglobin, right? However, in case of muscles, what happens if oxygen remains unused? So the amount of oxygen that is coming into the cardiac muscle or any muscle that has got myoglobin, जिसमें myoglobin है, वहाँ पे क्या होगा? The myoglobin, the oxygen is getting dissolved getting used up for ATP synthesis, and the remaining oxygen immediately binds with myoglobin to form myoglobin, oxymyoglobin, right? So whatever oxygen is coming to the heart, it is being fully utilized. The main function is production of ATP via cellular respiration or OXPHOS, oxidative phosphorylation. And even if some amount of oxygen is left, it will be immediately captured and will be stored as reservoir via formation of oxymyoglobin. Okay, these are very easy to understand, not a difficult concept. So when will myoglobin release oxygen? Okay, of course it will release, definitely, otherwise it is why it is there. Okay, it is a hemoprotein, its function is to release or provide oxygen. So it will only unload oxygen under extreme condition. Extreme condition means when it reaches very low, from below 5. So since the P50 is 2 to 3, remember P50 is 2 to 3. So by the time it reaches 5, almost 100% of myoglobin is already saturated. So when the partial pressure of oxygen inside the cells reaches as low as 5 millimeter of mercury, can we imagine oxygen's saturation 5, patient is almost gone, right? Even in those cases, the heart will continue to beat. That's why the first organ that is most vulnerable to this lack of oxygen is brain. That's why in case of when a patient dies, generally brain death occurs first, but still the organs continue to function. So there comes the forensic or medico-legal aspects of organ donation. We are not going into those details, but know this, heart can survive for long. Even when you, most of you I think have seen chicken or mutton being cut, right? Even when the animal is dead, the muscles have have gone into rigor mortis, or it has finished movement, the heart will still continue to beat. It's because of this, right? Anyway, so of course myoglobin is there for releasing in extreme conditions. So myoglobin releases oxygen in rapidly respiring cells. The released oxygen is used up in oxidative phosphorylation. So the oxygen that now myoglobin releases when the PO2 is below 5 will be still used for oxidative phosphorylation and production of ATP only, right? Because in order to beat, muscle needs energy. So what is the applied importance of this? Apart from the functional importance, has it is there any more diagnostic importance? Yes, there is. So myoglobin present within the muscle cells. I told you it is present in the cytosol. So if anyhow the blood, the cell gets damaged, okay, the ruptured cell membrane will release all the contents of the cell, and then the myoglobin present in the cell will come out into the bloodstream. That's why myoglobin is generally very high. And I told you what is the main tissue where myoglobin is present, main muscle, that is the heart muscle. So any reason when the heart muscles are damaged, the main reason is acute myocardial infarction and heart attack. Okay. So acute myocardial infarction, myoglobin is the earliest marker of acute myocardial infarction. So it is found increased in blood, it is found increased in urine. Everywhere you will find myoglobin. Okay, so it is a marker of myocardial damage. Remember, earliest marker, even before cardiac troponin and everything, myoglobin will be high first. So the first sign of an AMI is high myoglobin. However, since I told you it is not only specific to cardiac muscle, it is a non-specific marker. Maybe there may be other reason. For example, in skeletal muscles also there may be some disease, muscular dystrophy, due to which myoglobin may be high. So a proper history of acute myocardial infarction along with myoglobin high is much more suggestive. For example, you find isolated myoglobin high in blood or serum, you will say, okay, the patient is suffering from acute myocardial infarction, heart attack हुआ इसको नहीं, वो चल फिर रहा है. Basically, if the patient gives a history of chest pain, profuse sweating, etc. along with myoglobin high, then you can suspect it is a case of AMI. Of course, you need to confirm your diagnostic by doing troponin T, etc. There are multiple tests for acute myocardial infarction, but a proper suggestive history along with high myoglobin extremely points out towards acute myocardial infarction. So myoglobin is thus a marker of AMI, acute myocardial infarction, INFARCTION, not infection. Okay. So I told you when myoglobin is somehow, okay, the myoglobin iron is ferrous iron. That ferrous iron, under similar condition, when it is exposed to excess oxidative stress, etc., can be converted to ferric form. When it was happening in hemoglobin, it was known as methemoglobin. If the same thing happens to myoglobin, it's known as metmyoglobin. Extremely rare, because myoglobin is less in the first place, so metmyoglobin is also less. Naturally, the same thing will happen, it is non-functional form. Then metmyoglobin does not bind to oxygen. Okay, it will not be able to store oxygen as a reservoir in case of cardiac muscle. So remember, oxidative stress or increased exposure to oxidative drugs is not only a problem for methemoglobin in case of skeletal muscle, it is also a problem for cardiac muscle. However, I told you it is reversible. Excess oxygen, positive pressure oxygen ventilation will reverse the metmyoglobin and methemoglobin into their ferrous form. Okay. So what are the differences between hemoglobin and myoglobin? Okay. So let us see. So hemoglobin, so just you can take a minute, you can recap and then you can see at this slide, right? So see, hemoglobin is what does it do? Okay, it helps in oxygen transport in red blood cell. Myoglobin does not help in transport. It is a storage form. Okay, it's a reservoir. So oxygen storing protein in muscles. Okay, hemoglobin transports in blood, myoglobin functions in muscle. Hemoglobin transport, myoglobin stores. So two points you get in point number 1. Okay, hemoglobin can bind with 4 oxygens, since it has got a tetrameric structure. Okay, 4 globin chain. Myoglobin is monomeric, it has got 1 heme, and bind with 1 oxygen, right? Hemoglobin is oxygenated at the lungs. Definitely, since it's a part of blood is getting circulated in the pulmonary circulation, it is getting oxygenated at the lungs. Myoglobin is oxygenated at muscle cell cytosol. Myoglobin is inside the muscle, okay? Over here, after supplying all the tissues, the blood is reaching. So, oxygen tension is much lower compared to that of lung, right? So, myoglobin has to get oxygenated at a very low PO2 level. Hemoglobin unloads oxygen at tissues when PO2 is 40, okay? So, when the partial pressure of oxygen is 40 millimeter of mercury, hemoglobin will start giving out all the oxygen. However, in case of myoglobin, we just learned that it needs much more extreme condition to release, that is 5 millimeter. That too, only it will release to the cells, muscle cells that are actively respiring, those who are actively functioning, right? So, mainly cardiac muscle. So, P50 for HbA1 is 27 torr and P50 for myoglobin is 2 torr. So, you can see even at extremely less concentration of oxygen, myoglobin is very much saturated compared to adult hemoglobin. Regarding oxygen dissociation curve, it is sigmoid shaped, the classical sigmoid shape we know, S shape, okay? And in case of myoglobin, it is hyperbola or rectangular hyperbola, same thing. Not same thing, basically rectangular hyperbola means it's symmetrical up and down. And regarding molecular weight and the number of amino acids, since it is tetrameric, considering all the limbs, 574 amino acid and 67,000 Dalton or 67 kilodalton. And in case of myoglobin, it has got 153 amino acid in its one globin chain and 17,000 Dalton or 17,200, 17.2 kd. Okay, so this is the difference between hemoglobin and myoglobin. So, the chapter is over, okay? We will now look at the questions. Okay, so you can note down the question, you can take a screenshot. Basically, these are the questions that needs to be covered from this whole competencies. So, let us see whether we have discussed all, and if you have any problem in any question, feel free to get back to me personally and I can help you with either this video or I will share the links or I will again teach you, no problem. So, structure of hemoglobin. Okay, the tetrameric structure, structure of heme, structure of globin, very important. Function of hemoglobin and biomedical importance of hemoglobin. Okay, oxygen carrying, oxygen loading, oxygen unloading. Okay, everything should come as a long question. Next, salient features of hemoglobin, oxygenation, deoxygenation. Okay, how taut form, relaxed form to everything happens, cooperative binding, all important. So, allosteric effectors of loading and unloading of oxygen by hemoglobin. Okay. 2,3-BPG and its role in hemoglobin, discussed in much detail. Oxygen dissociation curve of hemoglobin and the factors that affect, discussed. Carbon dioxide transport in human body, okay, carbaminohemoglobin and via the bicarbonate buffer, those are also very important. Next, normal hemoglobin variants, embryonic hemoglobin, fetal hemoglobin, adult hemoglobin, A1, A2, right? Glycosylated hemoglobin and its significance, HbA1c, what is the importance, how it plays a role in diabetes, okay? Hemoglobin derivatives, right? So, normal and abnormal. Dyshemoglobins, abnormal hemoglobin derivative, okay? Carboxyhemoglobin, methemoglobin, right? So, basically these are the same thing, these are various questions that came in previous years. So, when we are discussing about hemoglobin derivatives, we can discuss about abnormal hemoglobin. When we are discussing about abnormal, we are going to discuss about carboxy methemoglobin anyway, right? So, type of hemoprotein we discussed in the very first lecture, hemoglobin, myoglobin, cytochrome, etc. So, type of hemoproteins are essential. There are few differences that you should be knowing as well. If you have read the whole content in detail, definitely you will know the differences. So, hemoglobin A, hemoglobin F, okay, difference adult hemoglobin and fetal hemoglobin, difference between hemoglobin and myoglobin, right? these are the slides I think I have showed you in different form anyway. So the T form and R form of hemoglobin, okay. Hemoglobin at lungs and hemoglobin at tissues. Basically, oxygenation and deoxygenation of hemoglobin, or oxyhemoglobin or deoxygenated hemoglobin. These are the few differences that you should be knowing. All right. So I thank you all for your patient hearing, and it's over. The class is over for the first time in a long time. I have finished it so early, but okay. So okay, I'm glad, and I hope that this lecture series was very detailed but was very helpful. Okay, and feel free to get back to me with any query. But I have something to show you. You'll be surprised to see the number of slides. Okay. Generally, when a teacher teaches you, you can guess, okay, number of slides that he or she will be showing kitna ho sakta hai maximum, because one slide actually takes 5 to 10 minutes to explain, if you are explaining everything in the slide, simply by not disrespecting anybody specific, but there will be many will be simply reading the slide. Samjho na samjho kya fark padta hai, right? But if my goal is to make you understand, अगर समझाना है ना तो टाइम तो देना पड़ेगा. समझते हो कि नहीं समझते हो, that is ऊपर वाले के हाथ में है, but at least I should give my effort. So in a 60-minute class, okay, even if 5 minutes per slide is allotted, so कितना slide हो सकता है maximum? 12, okay, 15, ठीक है. Okay, if वैसे स्लाइड्स हो जो मतलब चेंज करो रीडिंग मैक्सिमम 20 स्लाइड शुड नेवर बी देयर इन अ 1 hour lecture series in either lecture hall or in online session. Okay, that's a tip for any teacher who is watching this video. However, I actually designed this slide keeping all the series in mind. So there are, can you see the number? 471 slides है यहाँ पे. So अगर if you can calculate, then it will take so many long time. But the reason why there is 471 slide, because एक slide में there is only one line. Okay, so a very important tip for teachers. Okay, whatever you do, okay, now students, you will also be preparing student seminar in future. ठीक है, will also be you will have to teach anyway. In order to learn, you need to teach. As simple as that. अगर नहीं पढ़ाओगे तो सब भूल जाओगे. There is a reason. Okay, there is a reason why good teachers are good teachers, because they love to teach. Anyway, I am not talking about myself. So देखो, always this is a rule. In one slide there should not be more than 6 lines. Okay, 6 lines से ज़्यादा नहीं होना चाहिए एक slide में. And in a single line there should not be more than 6 words. You get my point? When you are preparing your own slide, make it a point, not more than 6 lines in one slide and not more than 6 words in one line. So this is the rule of 6 by 6. अगर ऐसे होता है, if this is so, then only the person that is sitting, or the student that is sitting in the last bench, will be also be able to view this. Because being a teacher, it's very important so that every student sees your lecture. कुछ ना कुछ तो याद रहेगा ना, अगर मन नहीं दे रहा फिर भी कुछ ना कुछ याद रह जाएगा. ऐसे स्लाइड्स बनाएंगे तो ही याद रहेगा. Anyway, बहुत सारा लेक्चर दे दिया. I thank you all for your patient hearing. Soon we'll be starting with another chapter, another topic. But till then, bye and take care.